previous lecture we was discuss about the sexual reproduction and the types of sexual reproduction, and today we will discuss about the types of asexual reproduction. Asexual reproduction, that means only single the type of reproduction in which only single parents is involved. It is further of many types. First is fission. Fission means breakdown. When the parent cell, the nucleus of the parent cell divide into two or more than two parts, it is known as fission. It is further of two types. First is binary fission and the second one is multiple fission. First we will discuss about the binary fission. In binary fission, the parent cell, first the nucleus of the parent cell divided into two nuclei. That process is known as karyokinesis. And further the cytoplasm divides. And this division of cytoplasm is known as cytokinesis. After the result karyokinesis, then cytokinesis, two daughter amoeba or daughter cells are formed. If we draw the diagram of the amoeba, this is its nucleus, this one cytoplasm, and that are plasma membrane or cytoplasm. First, nucleus elongate and divide into two parts. At the same time, when the division of the nucleus is almost completed, the same time the constriction of the plasma membrane at that part gets started. To divide the cytoplasm. This one is our nucleus, two daughter nuclei, and finally two daughter amoeba are formed. So this one is our parent cell. First the karyokinesis, nucleus division takes place, and at the meantime cytokinesis takes place first by the constriction in the plasma membrane, and finally two daughter cells are formed. The next type is multiple fission. Second type is multiple fission. This is very common in malarial parasite, that is Plasmodium vivax. In Plasmodium, The development of the this malarial parasite takes place in the red blood cells. This is our RBCs or red blood cells. Nucleus divided into firstly two parts, then 2 to 4, 4 to 8, and multiple division takes place in the nucleus. When the division of nucleus takes place in the much extent, it exerts pressure on the wall of the RBCs, and finally the wall of RBCs get ruptured, and these nuclei comes outside. Ruptured wall. Our next type is, this is all about fission. The next type is budding. Budding takes place in hydra, in animals, and yeast in budding. A small outgrowth, if we take the example of hydra, a small outgrowth from the parent cell arises. This outgrowth increase in size when this outgrowth increase in size and fully grown, it detached from the parent plant and dwells the darker height during the favorable condition. Our next type is spore formation. Which takes place in fungus. फंजाई में होती है. इसमें जो बॉडी होती है वो माइसीलियम डेवलप कर देती है, सबस्ट्रेटम के ऊपर माइसीलियम डेवलप कर देती है, दैट आर फॉल्स, जिससे ये सबस्ट्रेटम के ऊपर अटैच होता है. The stalk-like portion where the stalk-like portion where the spores are formed is known as sporangium. And fully grown, this is fully grown sporangium. That is stolon, rhizoids, sporangium 4. This is mature sporangium. स्पोरेंजियम। जब फंगस में ग्रो करना होता है तो ये बेस के जिस सबस्ट्रेट के साथ अटैच होते हैं, ये अटैच होते हैं थ्रू द राइजोइड्स। राइजोइड्स से अटैच होने के ऊपर ये स्टॉक लाइक जो स्ट्रक्चर्स बना लेते हैं, उनको बोलते हैं स्पोरेंजियोफोर्स। स्पोरेंजियोफोर के ऊपर होते हैं स्पोरेंजियम। स्पोरेंजियम के अंदर जो स्पोर्स की डेवलपमेंट होती है, जब स्पोर्स की डेवलपमेंट कंप्लीट हो जाती है तो इनकी बॉल बस्ट कर जाती है और स्पोर बाहर रिलीज हो जाती है। और ड्यूरिंग favorable condition these spores develop into the new organism. Next type is fragmentation. Which takes place in plants only. एग्जाम्पल है इसका स्पाइरोगाइरा एल्गी। स्पाइरोगाइरा is the algae. In this, the chloroplast is present in the spiral shape. This is a plant cell. इसके अंदर क्लोरोप्लास्ट जो होता है स्पाइरल शेप है। That's why this algae is known as by this identifying feature. प्रोमिनेंट न्यूक्लियस होता है, एक साइड के ऊपर प्रेजेंट होता है। This fragment of this is one cell of the spirogyra. एक सेल जो बीच में से कट हो जाती है, one cell of spirogyra divides into two cells or forming two daughter spirogyra. Our next example is regeneration. It mainly takes place in planaria, or it is only takes place in the animals. Planaria में क्या होता है? बॉडी है इसकी, ये इसकी आईज, इसके बॉडी इसकी 3 पार्ट्स में डिवाइड है। और तीनों पार्ट जो है न्यूली ऑर्गेनिज्म फाइनली डेवलप इट्स बॉडी और फॉर्मिंग न्यू ऑर्गेनिज्म। तो एक प्लेनेरिया सेल में कितने ऑर्गेनिज्म मिले? 3 ऑर्गेनिज्म। This is about regeneration. Now the vegetative propagation is also considered a type of asexual reproduction. Vegetative propagation. अब वेजिटेटिव प्रोपेगेशन होता क्या है? द फॉर्मेशन ऑफ न्यू प्लांट्स बाय द रूट्स, वेदर बाय द रूट्स, स्टेम और लीव्स ऑफ ए प्लांट इज नोन एज वेजिटेटिव प्रोपेगेशन. इट इज फर्दर ऑफ 2 टाइप्स. फर्स्ट इज natural vegetative propagation one is artificial. Vegetative propagation. In natural vegetative propagation, it takes place whether by the roots, stem or leaves. नेचुरल मतलब नेचुरली वो अपने आप प्रोसेस हो जाता है, उसमें मैन को कोई एक्टिविटी की नीड नहीं होती है। मैन को कोई भी एक्टिविटी की नीड नहीं होती है। एग्जाम्पल रूट्स के लिए है स्वीट पोटैटो, स्टेम पोटैटो और लीव्स में होता है ब्रायोफिली। Artificial vegetative propagation is layering. For example, in strawberry. In this type, the main branch of the plant turns into the soil or bends towards the soil and touch the ground, and that is its taproot system. After touching it, after some time during favorable condition, it makes its own roots and small new plant is grown, which further develop into new plant. The second type is grafting. It occurs in banana, lemons or any other fruit trees. In grafting, small portion of one plant with desirable stem, that is known as scion, and another half portion of another part with desirable roots, that is known as stalk. They both are tightly binding with cloth or polythene paper. After some time, after few days, the cellular material, the tissue fluid exchange with each other and forming new tissue culture. Example, dahlia, orchids. Small tissue, desirable tissue of the plant is taken into culture medium, which is rich in nutrients. After some time, this tissue develops and forming a mass of cells that are known as callus. This callus is then transferred into another petri dish with nutrient medium, fresh nutrient medium and with vitamins that then or hormones. Then this develop first desirable foods, then this callus with roots, small roots, is taken out from that petri dish and converted into the another petri dish, which another contain fresh medium of nutrient and other physical conditions that are desirable to grow its shoot parts, and finally it develops its shoot or small stems. Then these small seedlings are converted into the main seed or converted into our main plant. So this is all about the artificial propagation. What are the advantages of vegetative propagation? It is easier method, cheaper method, and the fruits the plants are obtained in less value and not very costly, or genetically भी similar एक दूसरे से होते हैं. So this is all about the asexual reproduction. Thank you.